Assalamu alaikum to all viewers. Welcome to my channel. In this video, we will discuss about the DNA and molecular hydration. First of all, we will discuss about the DNA structure, and then we will discuss about the molecular hydration. You can first of all see here the A concept. DNA has a regular structure. Its orientation width between nucleotide length and width of nucleotide per helical turn is constant. All of these features were described by Watson and Crick. Adenine is always opposite thymine, and cytosine is always opposite guanine. The two strands are held together by hydrogen bonds. Two bonds between adenine and thymine, and three bonds between guanine and cytosine. I can scroll it, and you can first of all see here the concept. The DNA is a string of deoxyribonucleotide. These consist of three different components. These are deoxyribose sugar and a phosphate group, and a nitrogen gas. Variation in the nitrogen gas composition distinguishes each of the four deoxyribonucleotide. Also, you can see here the basic building is the deoxyribose bose sugar. This sugar is distinguished between because it contains a hydrogen atom at the number 2 carbon. Normal ribose has a hydroxyl group at this position. Attached to the 5 carbon is a triphosphate group. This group is important because in a DNA chain it undergoes a reaction with the 3 OH group to produce polydeoxynucleotide. The final feature of the molecule is a nitrogen base. These are attached to the 1 carbon. Four bases are possible: two pyrimidine and two purine. The double standard DNA molecule is held together by hydrogen bond. Pairing involves specific atoms in each base. Adenine pairs with the thymine, and guanine pairs with the cytosine. These pairings and the atoms involved are shown to the right. You have probably heard of ATP, the energy molecule. It is to which adenine is attached. This molecule serves two very important functions in biological organisms. The structure of deoxyribonucleotide and base pairing among n bases. Also, you can see here a single strand molecule of DNA. Each strand of the double standard DNA molecule has the same basic structure. It is a series linked together by phosphodiester bonds. Also, we discuss the detail about here. New algorithm for DNA sequencing by hydration. I can scroll it, and you can see here DNA sequencing. This is not an easy task because the two DNA sequences of a DNA molecule are usually so large that they cannot be read in one piece. In 1977, 24 years after the discovery of DNA, two separate methods for DNA sequencing were developed: the chain termination method and the chemical degradation method. Later in the late 1980s, an alternative and much faster method called DNA sequencing by hybridization was developed. DNA sequencing by hybridization works roughly as follows. The first phase of the method consists of a chemical experiment which requires a so-called DNA array. A DNA array is a two-dimensional grid whose cells typically contains all possible DNA strands called probes of equal length. L, for example, consider a DNA arrays